Welcome to the Health and Human Services Finance and Policy Committee for Tuesday, March 3rd, 2020. We do not have a quorum yet, but members, I am really excited. The Ag Committee gets to talk about farming, and the Environment Committee gets to talk about mining and forestry, but we have a really important business that has started and flourished here in Minnesota, and they're represented by Medical Alley. And because we make so many healthcare decisions, I wanted us to hear some of their stories so that we are not making decisions in a vacuum. And with that, I want to welcome Bobby Patrick to introduce us to Medical Alley, some of what they do, and then walk us through some of the amazing stories that they provide to Minnesotans and patients around the world. So welcome to the committee.
Thank you, Madam Chair, Senators. My name is Bobby Patrick, and I am the Vice President of Strategic Growth and Policy for Medical Alley Association. Thank you for the opportunity to be here today and provide a brief introduction to what you will hear and see. The mission of Medical Alley Association is simple. We champion and facilitate an environment that enables Minnesota's health, technology, and care organizations to innovate, succeed, and drive the evolution of healthcare. Our work, such as the Healthcare Transformation Initiative, brings together leaders from across Medical Alley to identify ways that sectors can collaborate to initiate meaningful improvements to healthcare. We have taken on this work because there is no place on earth like Medical Alley. Patients come from around the world to be treated here, and the products researched, designed, and developed and manufactured here improve lives on a global scale. This is the birthplace of implantable medical technology, collaborative care delivery, and innovative health plan models, all of which make care more effective and available to more people. More than 500,000 Minnesotans are employed by healthcare companies located in Medical Alley, and these companies collectively have an economic impact on Minnesota exceeding $60 billion. Today you will hear about some of the amazing work being done by some of these very health technology organizations that we are fortunate to work with every day. The innovations they and others are developing and deploying will change healthcare for the better for everyone. The challenges they faced and are facing in bringing their work forward to benefit patients is very real. It takes time and resources to get them into the hands of researchers, doctors, and most importantly, patients. We look forward to hearing the stories from these 4 innovative companies today and to continue sharing the insight, ideas, and innovations from our members that will continue increasing access, reducing costs, and improving outcomes for patients across Minnesota and throughout the world. And with that, Madam Chair, I'd like to introduce Jeff Ross from Miromatrix.
Welcome, Mr. Ross. Thank you for being here. Please state your name for the record and begin your testimony.
Yeah, hi, Madam Chair. It's Jeff Ross. I'm CEO of Miromatrix Medical. Madam Chair, members of the committee, it's a delight to be here and just kind of share some of the stories of a biotechnology life science company that was founded here at the University of Minnesota, and that we continue to move forward with a very grand mission. And our mission is to solve the organ transplant shortage through developing transplantable organs. What I want to do today is just kind of give you an overview of who we are, talk through some of the stuff, but some of the challenges that we've done along the way to give you a little bit of a sense from the time of startup all the way through clinical trials. What does that really look like for a product of this nature that could have a major impact on the healthcare system, but certainly the economy as well? So if we look at this, Miromatrix is developing a revolutionary solution to solve the organ transplant crisis and eliminate the waiting list. We essentially are changing medicine. Our approach is based on technology that was developed at the University of Minnesota, and this is called perfusion decellularization. And that's the ability to take a whole existing organ and we actually cannulate it, meaning we hook a tube up to it, and we perfuse a mild detergent through it. What that allows us to do is wash out all the cells. Kind of describe it like remodeling your house. If you think of drywall as the cells inside of an organ, we essentially remove that. What that leaves you with is the perfect scaffold, and I'll show you some images of that in a second. We're then able to place that into a bioreactor, which you see in that middle column there, and then perfuse human cells back into it and essentially reanimate that organ to be able to create transplantable organs. We've done a ton of de-risking. We actually took that first base material, which is a whole porcine liver, perfusion decellularized that, and we created medical devices with that. We launched 2 products, one called Miromesh, one called Miroderm. Got those through the regulatory process, manufactured those in Minnesota, and those are being transplanted into humans today. That's the base material that we're going to build our human organs off of. We have a lot of IP. I'll show a slide in the end. A lot of cost goes into developing an IP strategy, but more importantly, getting patents and being able to protect those patents as well. If we look at our programs, what are we trying to develop? Our lead program is a transplantable liver. Large market opportunity, just within the U.S., is estimated over $10 billion. And then also to go after kidney and create transplantable kidneys. I'll talk about that in a second. To date, as an entity, we've raised $47 million. Our last round was a $16 million round that we also brought in a large strategic. Currently out fundraising right now for an additional $40 million for the Series C, which will bring us all the way through enrolling 5 patients into our phase 1 clinical study for a transplantable liver. Those are our plans. We're targeting that in 2022. Just to give you a sense of, if we're looking at something this grand, how much capital does it really take to be able to bring a product like this to market? Knowing that within medical devices, there's a large continuum, right? Some need relatively small amounts, some need larger amounts. So just to walk in a little bit, what are we trying to solve? If we look at this is a nice little chart here of just showing the number of patients today who are waiting for an organ. In America today, there's 115,000 patients. Each year, 80,000 patients do not receive an organ. So if we look at kidney alone, there's 95,000 patients who are currently on the list waiting for a transplantable organ. There's about 21,000 kidney transplants performed in 2018. The average wait time is 3 to 5 years. More importantly, I think the stat that really talks to itself is that ability that each day 13 patients die while waiting for a kidney. Another 8 patients are actually usually brought off the list because they're no longer healthy enough to qualify for the surgery to actually get a transplant. Transplant outcomes are far superior to dialysis. Similar with liver, the 17,000 patients are currently waiting on the transplant waiting list for a liver. There's no alternatives to if you're an end-stage liver failure, and there's about 40,000 deaths associated with that. So there's no drugs, there's no dialysis, there's no devices. So unfortunately, either you receive a transplantable liver or you really don't have any options. So if we dive into this a little bit, I won't spend a lot of time on this. I mentioned liver's our lead for that reason, that there's really no alternative therapies that exist out there for liver therapy. And what we're planning to do is develop a transplantable liver to be able to solve that need. Where are we to date? I'll talk a little bit about the timelines that were associated with that as well. But we've already met with the FDA. We had our Interact meeting, so that's the pre-IND. That's essentially where we talk about all the data that's going to be required for us to get into human clinical studies. We believe that we're the only company that's been granted an Interact by the FDA at this point, just demonstrating how far ahead we are inside of that process. And again, we're planning our phase 1 clinical studies in 2022, and then targeting orphan status, just because there's no alternative therapies for it, in 2024. Kidney even gets more amazing by the number of patients and the ability to be able to affect these patients. If we look at today, there's about 500,000 patients in the U.S. who are on dialysis, with 100,000 patients annually added on dialysis as well. But I think what a lot of people don't know is the outcomes are incredibly poor with dialysis today. So if we look at it, 20% of patients die within 1 year, 50% of patients die within 5 years. Today, those outcomes are worse than many of the cancers that exist in terms of diagnosis. Renal care really hasn't kept up a pace innovation-wise compared to other sectors inside of our healthcare system, which is another thing that is helpful in the fact that an executive order earlier or last year on advancing American kidney health provides clear momentum. And they're really calling for, how do we invest in this sector? How do we create a bioartificial or an artificial kidney to really give these patients a new option? As part of that, they spun out an entity called KidneyX. It's a private-public partnership, and we were one of 15 teams that won a phase 1 award. Why that's important, there was 165 teams that applied to win that award. And really the focus is on how do we create new renal therapies. So that's really what we're going after and why we're trying to create this. So how are we going to do this? I talked a little bit in the beginning just to give you a sense on what our technology looks like. And that's the ability to be able to take an existing organ. We take them out of pigs because it's a byproduct of our food process today. There's an endless supply, and we can be able to create that pristine matrix. We take that, use our process to decellularize that, That allows us that first product where we then have that perfect scaffold. We then take organs that were unsuitable for transplant. So for each organ that is placed into a patient, there's multiple organs that don't make it, whether it's from cuts, tears, nicks, trauma. We're able to source those. Now we have good, viable cells. We're able to isolate those cells out and place them onto that scaffold to then be able to recreate that organ to essentially increase the supply of transplantable organ. That's our phase 1. Phase 2 product, I won't talk much about this, but that's the long-term potential, and that's the ability to be able to engineer organs from stem cells. So whether they're patient-derived or allogeneic stem cells, but that ability then to be able to create organs that no longer need immunosuppression, kind of the holy grail. But that's further inside of the future associated with that. So at a high level, that's kind of what we're doing. Grab an existing organ, remove the cells, place new human cells on it to be able to create that long-term solution. So again, at a high level, what's our technology? There's a pig liver there. You can see we cannulate that. We perfuse a mild detergent through it. By 24 hours, it's completely white. It's essentially called a ghost organ at that point. In this video right here, I'm just going to demonstrate that as part of this process, if we reperfuse this with a red solution, you're going to be able to see that all the vasculature is intact still. So we don't have that ability of how do you recreate that substrate and have all that intact vasculature. Again, all that architecture is there. So we now just need to recellularize it to be able to create that end solution. So if we look at this and we kind of parse this out, how do we get all the way to from where we are, inception, all the way through? We created 4 buckets that we had to demonstrate. First, prove that perfusion decellularization is effective. Then how do you recellularize it? Can we actually put cells back onto it? And can they engraft in the appropriate geometry? Third, can we then implant that into preclinical studies, demonstrate functionality associated with them? And then that allows us to work with the FDA and then move into human clinical studies, demonstrate the efficacy there, and now you have final approval. Those are kind of the big buckets as we look through the process. So where are we? We've demonstrated perfusion decellularization is effective. We created commercial product. That's our Miro Mesh, Miro Derm. Created a manufacturing process associated with that. Completed multiple clinical studies. So now we are demonstrating that that base material is incredibly effective. It's non-immunogenic. We're able to now build the substrate off of that. So the next step is, how do you now revascularize that? So how do you add cells back in and demonstrate that you can recellularize it? And I won't go into a lot of the data there, but we're able to source cells, grow those up, seed them back inside of the grafts. These are just a couple of images here, just demonstrating how this works. So this is a revascularized liver graft that's going back into a pig inside of this model. And what you're going to see is what this looks real time as we take those clamps off of it. This isn't overly bloody, so this should be fine. But what you can see, right, is now we're able to actually create a substrate. We're able to transplant that back in. When we take off that clamps, you can see that's essentially what a native liver looks like at that point. We then take off the other clamp. Now we've actually anastomosed back into the host, and we have the circulatory system going through it. So now we've demonstrated the matrix is okay. Now we've been able to revascularize this. And we can do this with a kidney as well. This is an angiogram after 10 days post-implantation, just demonstrating that we have that patent vasculature. So as I mentioned, we're able to take the matrix, we're able to add cells back onto it from the vascular standpoint, be able to transplant that back in. Now all of our focus is really on the ability of demonstrating in vivo functionality, of now adding the specific organ-specific cells to move to that next level. A lot of data on this slide, I won't go through it, but to demonstrate that, yes, we can now take liver cells, seed those back on, we can get liver function back out inside of our bioreactors. We also now are surgically implanting these and looking at functionality of those as well. So long-winded way of just saying things are going well. We continue to move in the right direction. And if we look at timelines from where we are today to where we want to be in terms of regulatory and risk, on the liver, we've really moved through the research phases. This gives you a sense of how you need to move through from the standpoint of de-risking the technology, proving that your value proposition is effective, and then moving into the development phase, which we are right now in terms of rescue of preclinical animals to be able to work with the FDA to be able to support IND studies, do the IND submission, which then would allow us to get into phase 1 human clinical studies. Now, for a product like this is a full BLA. We'd have to go through all the phases of clinical studies to get final regulatory approval from the FDA. In parallel with that, a lot of conversations go around reimbursement. What does reimbursement look like as well? To move all of those are working pieces that we continue to work through. If we look at the timeline associated with the kidney, very similar in terms of research phase, where we're at today, development phase, demonstrating that we can recellularize that whole kidney, place it back into a large animal model, and start to be able to bring back that renal function, which is that really big next step as we continue to target how do we create life-saving technology for dialysis. Won't spend a lot of time on this. Senior management, including myself, that was introduced, but board of directors as well, multiple individuals across the state, many of them current or ex-CEOs as well in the medical community. Just real quick on financing as we talk through this, just to give you a sense of what this looks like in the phases. So we were spun out of the University of Minnesota in 2009. In 2010, we initiated our first seed round that allowed us to create some of those early accomplishments. We then went into a Series A. Again, being able to then start to look at what that core patent looked like, protect the IP to be able to build a company around that. Then had a Series B, which allowed us to launch those first products and de-risk it. And then a B2 that allowed us to start getting some of that preclinical study that I just demonstrated to you. C, our next funding round, then sets us up to finish out those efficacy studies in our preclinical models and then move into human clinical studies. Have a very strong IP portfolio, so all 3 of our parent patents have now issued. We have 7 issued patents in the U.S. and covered in all OUS markets as well. This is just, I just threw this in here as a comparable. So what would this mean as we continue to go forward if we're successful in bringing the product all the way to the market? It brings tremendous value to patients, but also, I think, to the ecosystem and what we're trying to achieve here. So that's Miromatrix.
Thank you very much, members. For each of our guests today, we're going to take a little bit of time for questions. So my first question, I saw on the slide, phase 1. Could you walk us through just what the phases are for product development? And this isn't really a product. I don't know how to describe this, but FDA interaction.
Sure, Madam Chair. So if we look at, there's different classifications of products, right, depending on the risk profile, if they're class 1, class 2, class 3 with the FDA. Some of those can go through a regulatory process called a 510K, where essentially if there's an equivalent product that already exists on the market, you then can file and make the argument that your product is similar to that. What that allows you to do is void clinical studies and get that product approved, so then you're able to sell that product. For class 3 and products like this, there's some exemptions in class 3, but for biologic-based products like what we're developing, pharmaceutical products as well, you need to move through that full pre-market approval, PMA, to be able to then demonstrate safety and efficacy for the FDA to give you final approval of those products. So that's usually phase 1 is usually a safety study. So that'll be a small study in pharmaceuticals. That's your dosing study where you'll apply different doses and look for safety. Phase 2, you start to get into some level of efficacy to be able to look at, does your product work? And then in phase 3 is usually your pivotal study. So that's where you actually power it to demonstrate that, yes, it's having the intended effect that you have, and that's where you'll get your value props. With biologics like what we're developing and what you're starting to see with genetic gene therapy that's starting to come out, now you're starting to see those first approvals in some of the cell therapies called CAR-T that are going after some of the non-tumor cancers. What you're seeing is much shorter phase 1 and phase 2 and smaller pivotal, because it's a lot easier to demonstrate efficacy. What I mean by that is that if a patient doesn't have a gene, in your phase 1, you're already proving efficacy because you can quickly test, do you have that or not? Similar with ours, as we look at our product going forward, when we do liver transplants and we do kidney transplants, even during the safety study, you're gonna know, do you have function or not? So that allows that to shorten that window somewhat.
Excellent. Thank you. Senator Jensen, did I see you?
Thank you, Madam Chair, and thank you, sir, for your presentation. I was just curious about when we take the scaffolding, if you will, from the pig and wash it and then put the human cells back in, and you get the product and to the end point where it's ready to be transplanted. How does that compare to some of the research where we're actually growing the human organ in a pig. We take, and that, how, in terms of a total price before you go, and then also outcomes in regards to successful transplantation without rejection. Thank you.
Right. So, Madam Chair, Senator
Doctor.
Jensen, great question. So for the first one, this is a huge area of development, right? True need. I mean, there's tons of organs that need to be developed. So there are a couple of technologies. Ours is one approach, the ability to take a matrix, add human cells onto it. Our approach really looks at, if we look at from the immunogenicity side, since we're recellurizing with human cells, we anticipate that we know what that immunosuppression is going to need to be able to do, because it should be similar to cadaveric organs or living donor organs today. There's another approach, and that is, well, can we just genetically modify the pig to be able to take that animal, to take that organ directly out of the animal and place it into humans? So that's called xenotransplantation, and there are multiple companies working on that approach as well. Probably been working on it for the last 30 years. Originally, they got hung up with a couple of proteins called alpha-gal and some other ones that we have a ton of antibodies to. So the problem is you can't take an animal organ, put it in a human, because we immediately reject it. So they are using current technologies such as CRISPR and gene modifications to be able to modify that genome to see, can we get it to the point that you can transplant it into a human? And, you know, I think the verdict is still out. I think they're up to 15 or so genes that they're currently knocking out to be able to try to get there. But the open verdict, I think there, the unknown is even once you get past the immunogenicity, A, can you get past it? But B, from a functionality standpoint, will it function the same, right? Body temperature is a little bit different. Some of the receptors are a little bit different. And I think that's still the unknown on that. Where on our approach, as we talked about, right, we've already de-risked the porcine scaffold once it's decellularized, because our current products to de-risk and take that immunogenicity off the table, essentially, and then add human cells onto it.
Thank you. Thank you very much for your presentation today. Truly impressed, and given how much we spend on dialysis, it'll be interesting to see how your approval process goes, and then how we get this into our market. So I thank you for your time today. Oh, Senator Rosen.
I'm so sorry, Madam Chair. Thank you. Mr. Ross, is this a very competitive market? And if it isn't, the development of different techniques, or explain to me, is it competitive?
Mr.
As far
Ross.
as, do you have a niche on the market, or is there different?
Yeah, Madam Chair, Senator Rosen. Competitive, I would say no, because we don't have any therapies that are working today.
Okay.
Right? So there's no existing artificial kidney or bioartificial liver today to be able to solve that need. Is there competition in development of them? Yes, there's more today. But, you know, one of the challenges is expensive, as you saw, right? We've raised $47 million. We're going after additional $40 million. So you got to make sure that you have the correct investor base and technology to get that investment and continue to demonstrate the viability of the product to be able to continue that to where it needs to be.
And Madam Chair, just
Senator
one more,
Rosen.
just out of curiosity, Mr. Ross, these pigs are raised in a biosecure facility, I would assume. Is that correct?
Dr.
Madam
Ross.
Chair, Senator Rosen, no, in that, not in our process.
Okay.
So what we demonstrated inside of our process is, again, what we're using organs that were naturally discarded, right? So each day, because of a byproduct from the food industry, there's thousands and thousands of livers and kidneys that aren't utilized. We're able to actually source those. We have a couple of constraints by the FDA that we've talked about, but not large constraints, because our process we've already validated, demonstrates we get rid of the DNA, we get rid of the cells associated with it, and we get rid of the detergent and any viruses that may be inside there. So we've really validated already with the FDA that we can source that kind of common material, and our process removes any risk associated with it. Where, you know, what I was talking to Senator Jensen about, the transgenetic, now those do need to be in a high health, essentially a bubble, remove the viruses. So there's a second issue associated with that pathway on that other type of product. Yep.
Thank you, Dr. Ross.
Thank you,
Mr.
Madam Chair.
Patrick, who do we have next?
Next is Grady Hanna and Tom Burke from Nightwear.
Hi, welcome to the committee. Please
Thank
state
you.
your name for the record and begin your presentation.
Madam Chair, members of the committee, my name is Grady Hanna, and I'm CEO of Nightwear, and I'm joined by Tom Burke, our company president. Nightwear is it's a great Minnesota story. A brief overview, Nightwear is an AI platform and digital therapeutic that is delivered through the Apple Watch that treats nightmare disorder, which is a key symptom of PTSD. So one of the major symptoms of PTSD is re-experiencing of events. And many people with PTSD, they're re-experiencing these events in the form of a nightmare. And so, for instance, we've received breakthrough status from the FDA because people with nightmare disorder are, depending on the study, are 3 to 5 times more likely to exhibit suicidal behavior. And so early on, we connected with a medical device expert who advised us to go the FDA-regulated route and seek a reimbursable route. Just one example of the rich ecosystem that we have in Minnesota. And so the way Nightwear works is the wearer wears the watch, the sensors from the watch upload to our AI platform. That profiles and calculates a person's sleep. We can then detect when they're entering a nightmare, and we intervene through buzzing the watch to arouse them out of the nightmare without waking them. And we are in a randomized clinical trial at the Minneapolis VA. We are going to be submitting to the FDA for regulatory clearance in the next 90 days. Before I go further into Nightwear, though, I'd like to just describe a bit of how I see the medical device and healthcare industry in Minnesota, not just locally, but nationally. So having grown up in Minnesota, I'm a proud Minnesotan. I actually participated in youth and government here, and I've always had a real respect for the Minnesota government as a means of supporting systems that help many people. And I believe that if you look as a cautionary tale, Minnesota used to be the world center for supercomputing. We were the big six. We were Control Data. We were Cray. And we got outpaced by Silicon Valley. And so now Silicon Valley is the center of computing and innovation. When you think of course, St. Jude and Medtronic and Guidant, which is now Boston Scientific, of course they're extremely successful, but they're almost monolithic because, as you've heard here with the organ transplant technology, it's extremely specialized and very rigorous in its review. And so we kind of just view it as it just kind of exists. But it's created this foundation for companies like Nightwear. So in the case of Medical Alley, they proactively reached out to me. They have a means for startups to join free of charge. They reached out to me proactively, introduced me to my point of contact at the Minneapolis VA, who was responsible for us beginning our clinical trial, introduced us to our first lead investor, and continues to support us in multiple ways. And so when we think of the Minnesota as a national player with all the components we have with Mayo and the U of M and these huge players, I would just encourage you to realize that there's, and of course you do, but we have so many great pieces, and the more that we can cultivate that and create that and promote that capability, the better off we're going to be as a state economy. So in case, so kind of back to Nightwear and nightmare disorder. A young man developed the kernel of Nightwear while he was a senior at Macalester College. And I had lived 15 years out in San Francisco and had moved back, and I was interested in learning more about healthcare, and I read about him in the Star Tribune. His father had returned from Iraq with nightmare disorder and PTSD, and he invented this and was able to help his father and was promoting that fact, and I had learned about him. And so when I reached out to him, we were able to build a relationship and start the company together. And from there, we've built a robust AI platform that serves our veterans as well as other people with PTSD. And there is no current standard of care for nightmare disorder. So these are people that, in the case of PTSD, they can't even escape their demons in their sleep. And so it really puts them on the edge. And so to be able to treat these folks is really a privilege, and it's been an amazing journey thus far. People get very excited about the mission. So we've been able to recruit a senior management team, including Tom, and all people with extensive device and pharmaceutical experience. And there are 20 more people that have helped us in an ad hoc way, in a consultative way, that are local here in Minnesota. So once again, this long, rich history of more of the interventional care, right? Heart valves and stents. That builds a foundation of rigor, an institutional foundation, so that digital therapeutics, which is now a regulatory category within the FDA, we have a foundation to build on, along with many other modes of healthcare delivery. And so in terms of, there's a significant market. It's really kind of a misunderstood and poorly diagnosed condition. That there's a significant market opportunity, an opportunity to bring jobs and care to our citizenry. We're in a randomized clinical trial. We've been able to show efficacy and safety. We have a site here at the Minneapolis VA, and we're actually adding the Tampa VA, and are in discussions to work with Wounded Warrior as well. We've got a full commercialization plan, and our use of funds, because of the talent base in Minnesota and the fact that the cost of living is so reasonable compared to the coasts, we've been able to get to this point where we're 90 days from FDA regulatory submission for just over $1 million. So it's a very different profile than maybe what we're used to hearing about medical device. And it gives us the opportunity to leverage the talent here in all kinds of ways, and people that are investors that are savvy. So things like the investment, the angel tax credit, things like that really do make a difference. Pat Dillon with the SBIR does fantastic work reaching out to startup companies as well. So Nightwear is an AI platform, a software platform that's regulated and reimbursed as a device. So we're really at this neat, innovative intersection of where value-based care is going. And we couldn't be doing it in a better place. All things being equal, if the costs were the same in any place else in the country, I'd rather be building this company here. And so I want to thank you, Madam Chair and committee, for your time. And thank you for indulging me on my love for Minnesota and where I see this very important industry to our state's mind share nationally. People know Minnesota, but I think we can build on that narrative and really become more robust in how we promote Minnesota as a great state for health care and medical device delivery. And I think that the Medical Alley Group is doing a great job of that.
Thank you, Mr. Hanna. Senator Hayden.
Thank you, Madam Chair. Mr. Hanna, just a little bit about I don't want to get too far in the weeds, and don't tell me things you can't tell me, but how does it work? I mean, I know it shoots something up to your AI platform and then back, and then gently wakes people up or makes something happen to where they stop having their nightmare. But is that based on movement, or is it based on heart rate, or how does, in a very basic way, how does the device know that you're falling into a nightmare?
Sure, Madam Chair, Senator.
Hanna.
Those are the two main components, are heart rate and movement at this point. In choosing off-the-shelf technology, consumer off-the-shelf technology, we're able to utilize Apple's innovation curve. So body temperature, galvanic skin response, additional sensors as they come available will be instructive to our algorithm for this disease state and others. At this point, we're able to get a strong enough signal in the rapid change in heart rate associated with the nightmare and the movement associated, because more trauma-induced nightmares, people are no longer paralytic. They move, they thrash, they reenact the dreams, the sequences. And so we're able to get very, very good and strong signal through heart rate and movement at this point. And I do want to add,
Mr. Hanna.
excuse me, Madam Chair, the Minnesota Department of Veteran Affairs Support Our Troops program. We applied for and received a $100,000 grant from them. That was another key component of us being able to move forward with non-dilutive capital as we develop out our therapeutic.
Thank you. And we talk about mental health on this committee a lot, and especially veterans' mental health. And this is something that I hadn't ever heard of. And that it's coming to market this quickly, or not coming to market, but that you're getting it through the FDA process and a non-pharmaceutical way to help veterans and others with PTSD. Do you know, is this a device they would have to wear the rest of their lives, or are they actually being retrained to not have nightmares so that they can have steady state sleep?
Madam Chair, at this point, we don't have enough data to know whether we can change core brain function. What we find is that if somebody's utilizing nightwear for a period of time, and say they go on vacation or they don't utilize it for a few days, that for about a week, they're not experiencing the same level of nightmares, and then they do come back. Our hope is that after 3 to 5 years of data, we can see a change to their core brain function and their incidence of nightmares, and that's all biometric information that we're collecting along the way. In regards to mental health writ large, Learn to Live, Habit Aware, there are many great device and cognitive behavioral therapy companies focused on mental health, and there is, in general, Minnesota is a very proactive state around mental health, and so it's a great environment through the Manova Conference, through what NPR is doing with their mental health series. There are a lot of forums to discuss these types of issues and figure out where we have common ground to move forward.
Excellent. Thank you very much for your time today. Mr. Patrick, I believe Dennis Ware is your next
Thank you.
testifier. Thank you. And members, I'm sorry I didn't mention this earlier. All these slides are in your packet, so if you want to take notes, I know some of this is really technical and still a lot coming at us, but if you want to take notes so that we can have questions or follow-ups, the slides are in your packet. Mr. Ware, welcome to the committee. Please tell us your story.
Madam Chair, thank you for the opportunity to be here. I'd just like to tell you a little bit about the Nuvera story. I won't get too much into the science of the company, but I want to start out, before I do that, with just a little bit about my personal background and why I'm here. I grew up in Michigan. I ultimately went on and became a medical doctor. I was a cardiologist. I trained at the University of California, San Francisco. Then I went back to Michigan, where I practiced cardiology for about 10 years in Ann Arbor, Michigan. At that point, I got the entrepreneurial bug and wanted to leave medical practice and try and develop my own medical products. And at that time, I got my initial financing from a venture capital firm in San Francisco, where I had done my medical training. And when I was just about ready to start that company, two medical device executives here from the Twin Cities called me up, and this was in the year 2000, and convinced me that Minneapolis would be a better place to do this than San Francisco. And I still remember the look on my wife's face when I told her I changed my mind and we were going to go to Minneapolis instead of back to San Francisco, where she had relatives. But we did. We came to Minneapolis, and when I got here, those medical device executives, one was from Medtronic, one was from what was at that time called SciMed, went on my board of directors, and they became my mentors, for how to build my first medical device company. And I think that really is the key to what's great, and creates a medical device ecosystem here, is the ability of past people who have been here, who are willing to give back and try and teach other people that want to be an entrepreneur how to do it. And I never would have had success if it weren't for those people. Right now I'm on my third company in Minneapolis. My first company, which I started in 2000, was ultimately bought by St. Jude Medical about 5 years later. And my second company, which I started in 2006, in 2011 was bought by C.R. Bard. This is my third company. This company I did not found. The company was actually founded in St. Louis at Washington University, and then its initial early animal work was done in Seattle, Washington. When the company was about 4 years old, and up here is a little bio of the company called Nuvera, the company that I'm developing now. When the company was approximately 4 years old and they had really excellent animal data that they had developed, they decided to go recruit a CEO that had a track record of having successfully developed, these types of high-tech companies. They recruited me, and they said to me, if you come here and be the CEO, the investor said, well, we'll let you put the company in whatever city you want to put it. And so I moved the company from Seattle, Washington, to Minneapolis, where it's been since approximately 2013. And I still remember the look on the company, on the 12 or 13 employees that existed at that time in Seattle and the company, their face when I told them I was moving the company to Minneapolis, because it kind of looked about like my wife's face looked when I told her I was coming here. So anyway, we moved the company here. I've been here for 7 years now running this company. And as you see on this chronologic history, it's a long time and a long path to develop a true high-tech, what's called novel device that's never been done before. It's generally, in today's world, it's called a PMA track with the FDA. To develop this kind of a thing, generally, most people would say takes somewhere in the neighborhood of 10 to 15 years. And it's probably somewhere in the neighborhood of $150 to $200 million to do that type of a device. At this point, we've raised $150 million in this company. Last February, we raised our most recent round of financing, which was $79 million, which was designed to run our pivotal clinical trial. This is our fourth trial. We've done phase 1 trial, two phase 2 trials, and we're now in the middle of our pivotal clinical trial. What we do is we treat COPD, and this procedure is done through a flexible bronchoscope. You can see a flexible bronchoscope there going through down a patient's throat and into his windpipe. What the physician operator does is through that bronchoscope, he applies thermal energy, RF energy, to the right and left mainstem bronchus to attenuate the nerves that go to the lungs. And when you do that, attenuate these nerves, they're called the parasympathetic nerves, you are able to achieve a permanent generalized bronchodilation. Your airways open up. So in patients that have severe COPD or severe asthma that drugs are no longer successful for, you can go do this therapy and get a much more powerful effect for opening the airways. This is just a cartoon of our device, deployed in the left mainstem bronchus. The procedure takes about an hour to do. It can actually be done. Right now we're doing all of our cases as an inpatient, but the procedure can actually be done as an outpatient. And people go, well, why would that work? I mean, these nerves are there for a purpose. Why would that work? Well, medical literature is filled with the rationale for why it works to tap down these nerve effects. And in fact, trying to tap down these nerve effects is how the current drugs that are used to treat COPD and asthma work. These literally these inhalers that you see people taking that have these drugs that sell tens and tens of billions of dollars worldwide are actually done trying to block these nerves. The problem is that when you inhale these drugs through an inhaler, the drugs will preferentially go to the open airways, but not to necessarily the closed ones where you want it to go. So by going in and directly affecting the nerves directly, you can get a more robust effect from the denervation procedure. This is a very quick summary of our clinical trial, human clinical trials done over the last 7 years. I mentioned phase 1 trial, phase 2A, phase 2B. All of these studies are published. The most recent one, the Phase 2B trial, Airflow 2, was published as recently as November. It was that data that allowed us to raise the $79 million financing to run the pivotal trial, which is being done at 30 medical centers around the world right now as we speak. And why were people willing to give us $79 million to run this pivotal trial? Well, it's illustrated right here on this slide. This was a randomized, double-blind, sham-controlled trial. That means that both the patient and the doctor did not know whether they actually got the real therapy or whether they got a sham therapy. And by sham, we mean they got the bronchoscope put down, the catheter put down. The only thing that was different is the energy wasn't turned on. Randomized, double-blind, sham-controlled. And what we found out is that the patients that got the TLD, that stands for targeted lung denervation treatment, their adverse events, their recurrent respiratory events were reduced by more than 50%. And you go, what were those events that were reduced? Well, the patients that got this therapy had less recurrent bronchitis, less pneumonia, less shortness of breath, and fewer COPD exacerbations, following this procedure. It was this data that, as I said, that allowed us to go ahead and run the pivotal trial. The FDA said that the primary endpoint for our clinical trial would have to be COPD exacerbations. COPD exacerbations are what is called the holy grail of clinical endpoints for COPD. What an exacerbation is, it's when one of these patients who has COPD or asthma suddenly has a worsening of their symptoms that is so bad they have to go seek medical attention right then, and in some cases actually have to be admitted to the hospital, for more aggressive therapy like on a respirator and an intensive care unit. These recurrent hospitalizations are among the most expensive hospitalizations that happen in the country. They can literally, a week in an intensive care unit for one of these exacerbations can easily run $50,000 hospital bill. And what happened from our therapy on these exacerbations is the purple line up there on the right, and the graph on the right shows how we reduced recurrent hospitalizations in patients with COPD compared to the control arm, which is the gray arm. The graph on the left compares our therapy to reducing just moderate exacerbations, those kind that can be managed on an outpatient basis. But this was regarded as relatively stunning by the pulmonologists and the experts that deal with lung disease. If we are able to reproduce this result in the pivotal trial that we're running right now, this will be one of the really exciting stories in the medical device world. And we are approximately a year and a half from when we'll be able to turn over the card, so to speak, and see the data on the pivotal trial. Pivotal trial has 500 patients in it. And if you have any interest in learning more about that trial or anything, we have two websites. We have, the Airflow, it's the Airflow trial is the name of the study. There's a website for the trial. There's also a LinkedIn website. It's on your slide there for anybody that has more interest in this, but in learning about it. But that is what Nuvera does. Nothing like this has ever been done before. So, and this benefit that you saw here, to be eligible for the trial, you have to be on optimal drug therapy, meaning these are COPD patients that are on what's called triple drug therapy. And this benefit that I demonstrated to you is on top of optimal drug therapy. Thanks for the opportunity to be here.
Questions from members? So I saw there was a crossover point on the slides. Is that when this procedure is starting to not be as effective?
No, what that is,
Okay.
I'm glad you asked that question, because at 1 year, when these patients had been followed up for 1 year, the patients that had gotten the sham procedure, that did not get the active treatment, those patients were told after 1 year that they did not get the active therapy, and they were given the opportunity to then be given the therapy. Those are called crossover patients. So it turns out about half of the patients that were in the sham arm elected to say, okay, I'm really disappointed, but I'll take the procedure now. So the crossover began halfway through the trial, and so that's when patients had the opportunity to actually get the therapy, and the patients that crossed over have had the same benefit, that we've re-demonstrated in the crossover patients the same benefit again. Now that data has not been presented yet, but it will be presented this spring at the American Thoracic Society meeting in May. Will be seen for the first time. That's why I don't actually have a slide up here showing you what that data was, but that'll be coming out in May, which will add more excitement about this therapy when it does come out.
Excellent. Dr. Klein, sorry, Senator Klein.
Chair, thank you for a very fascinating presentation, and I'm restraining myself from asking all sorts of nerdy internal medicine questions. First of all, my concern is that if you stop people coming into the hospital for COPD, I won't have a job any longer. So,
I don't think you have to worry about that.
Typically, these are reported out as changes in pulmonary function tests. Do you have any data along those lines?
Yes,
Mr. Ware.
Pulmonary function test, FEV1, the standard spirometry test done on COPDs are all secondary endpoints in this trial, and we've tracked them in all of our patients. And the pulmonary function tests improve, along, with the therapy. From the FDA's perspective right now, they consider pulmonary function tests a surrogate endpoint. They really encourage you to track hardcore diagnoses, like exacerbations and bronchitis and pneumonia. But those are being tracked. But the pulmonary function tests improve also.
Anything further? So, is there a point at which these people will no longer need a drug? Will this be a substitute for medication?
No, this is, we're not reversing the disease here. What we're doing is we're trying to slow its progression. Drugs have never slowed the progression of COPD. They can make COPD patients feel better, but they just slowly keep getting worse over time. We're hoping that by reducing these recurrent exacerbations and pneumonias, that the patients won't decline as fast. They'll feel better, but the real win here would be if we can slow the progression, and then you would have the opportunity to maybe prolong life, if you do that. But you won't cure the disease.
Thank you, Dr. Ware. Oh, Senator Hayden.
Thank you, and thank you, Doctor, for coming over. This is really important stuff. My grandmother had COPD and lasted a little longer than most, but it's definitely a very tough disease. I just have a simple question. How often do people get this procedure? Is it a one-time thing or is it a regimen?
It's a one-time
Dr. Ware.
therapy. One time.
Okay. Thank you.
Thank you. Mr. Patrick, we have Ping Ye, I believe, with STEMonics. It's good to see you again.
Good to see you as well, Chair Benson, and thank you, committee members, for inviting me here today. My name is Ping Ye, and I'm the co-founder and CEO of STEMonics. And before I begin with the 2 to 3 slides that I have prepared for you today, I'd like to share the backstory on the creation of STEMonics. And it was really birthed through my experience as a cancer survivor. So 7.5 years ago, I was diagnosed with Hodgkin's lymphoma. And the first 8 rounds of chemotherapy did not work for me, and had to be given a more potent version of chemotherapy that was very toxic and more toxic than my previous regimen, and I nearly died from that treatment. And so when I was laying there in treatment, I often thought to myself one critical question and one critical problem, which was, how can we test medicines on human cells without risking the lives of the patient? And from that point on, STEMonics was born. Yeah. Oop. There we go. So this is STEMonics, Maple Grove. It does not look like this today on an early March day, but STEMonics, if you ask the industry how they see us, it's really as a company that has converged biology and software It's a company that it makes human organoids, and I'll talk a little bit about that, which is the ability to convert skin or blood into a form of stem cells. That won the Nobel Prize in 2012, and I was able to license that technology after my experience as a cancer survivor. I'm a nanotechnologist by training, and so I wanted to see if my background could be useful in the life sciences industry, and that's how the company was born. And I formed a team that really were on the forefront of stem cell technologies, and the main challenge was how can you industrialize these processes so that you could help the drug discovery industry test compounds that could potentially work for all of us. Currently, millions of animals are sacrificed a year to test if these medicines will work on people, but they are often not the right models, especially for neurological diseases. So StemoniX converges biology and software, and here's one of our products. This is a plate, international standard plate. It's filled with organoids of different formats, brains, hearts, pancreases, and they come in different formats. And once again, these do come from skin, so there's no embryos that are used to make this process. Now, we generate so much information from these organoids, and you'll see in a few seconds here what I mean by that. We need a lot of software to analyze that data. And so we're really seen as the leader in the world now in industrialized human organoids, and it's really maybe best exemplified by this picture. Every dot that you see here is a human cortical mini brain, as they say in mainstream media. Each dot is about 40,000 brain cells, and we brought on really the leaders in the field back even 10 years ago. The cover article of Cell was led by one of my lead scientists. And what was interesting here is once we actually made industrialized versions of these mini brains, we discovered a couple years ago that we could form, if this video plays here, you'll see a flash of orange light there. That essentially is an early form of brainwave in these mini brains. And we used these brain waves to test drugs, whether for safety or for discovery. We have the same for heart as well. You can see one of our mini hearts here looks just like the plate on the left, but they beat instead of communicate like the mini brains do in that video. To give you a sense of what can be done with this, if we plotted out this pulse at the top, you'll see this sinusoidal pulse that you can use for testing drugs like safety. So if that signal that you see at the top there deviates from normal, you know you've got an issue. And we're delving into ways of using that to determine what types of actual mechanisms of toxicity that are causing the issue with that drug. Now, what's really interesting with this technology is that this waveform that you see on the bottom is from a child with a neurodevelopmental disease. So what we're learning is that children or people with particular diseases and the brains that we make from them can actually have a fingerprint of their own waveform, thus giving us an opportunity to test thousands of compounds to figure out what of those compounds changes the biology to ameliorate the disease. And so here you can see at high scale, nearly 400 brains on a plate. You're seeing hours of data across all 400 of these brains. It's like an hour-and-a-half movie, if you will, in a single 400 hour-and-a-half movies in a single plate. And so we have to apply a lot of software, else it would take months by scientists to analyze this data. And essentially what we've done is shrunken the hundreds of hours down to 2 minutes using augmented intelligence, AI approaches, and data analytics. So that's really our advantage out in the marketplace, is this ability to make these organoids that are highly functional, that can screen thousands of compounds for the whole industry. Funding-wise, I'm very proud to be here because the state of Minnesota has been a huge part in the history of StemoniX. We were honored with the recognition of winning the 2016 Minnesota Cup and have been participated in the Angel Tax Credit Program. I think we were the top recipient back in 2016. And so it was a huge part of getting us to where we are today. And with regards to where we are today, I can say that we're making efforts to help even on a national level. So I'll skip over the fact that we use AI to find drugs and all of that. I don't know if we have time for that, but I can say that we were chosen by the National Institutes of Health as the brain model of choice to find painkillers that are less addictive in the national effort called HEAL, which is helping to end addiction long term. So they're using our brains to find painkillers that are less addictive. With regards to our traction, stemming here from Minnesota, we now serve 15 of the top 25 pharma in the world, serve government agencies, including the U.S. Army on fighting weaponizable viruses like Ebola. Our total paying clients now is 80 globally. And essentially, our products and services are helping our customers and partners save hundreds of millions, if not billions of dollars, which really gives us and them potentially the opportunity to address drug prices by giving them the opportunity to more efficiently get drugs to market. I'm very happy to say that 2 top 10 pharma companies will be presenting and submitting papers in peer-reviewed journals in the coming weeks. Here, a snapshot of some of our customers. Half of these we work with on a weekly basis. And I think just to tie up the ends here, we have been able to still be on the forefront of the leading journals. This was the October issue of Cell Stem Cell. One of my leading scientists was able to make a more complex version of brains that you see on the left there that has a subcortex. And essentially, this is also made from skin, but the electrical activity of this organoid had the similar to an EEG signal of a 35-week-old premature infant. So these are ways we can test drugs without actually risking the lives of people and children. Essentially, if I were to summarize everything that I've said today, StemoniX is essentially at the convergence of stem cell technologies, human organoids in one area, software analytics and AI in another, and the ability to screen thousands of compounds, including the ability to program or gene edit these cell lines. And so in summary, together we believe we can unlock some of the most challenging diseases that face society today. And here's a snapshot of the team. I'm not much without them, and it's an honor to be here with you today. Thank you very much.
Thank you, Mr. Ye. Senator Kiffmeyer.
Thank you, Madam Chair. This is an amazing hearing to be able to know about some of these phenomenal things. So thank you very much for being here and telling us about StemoniX, rather. So I have a couple things. One of the things that I take a look at, especially in your brain thing, demonstration material here, and that is, have you done any work on seizures and vaccines?
Chair. Oh.
Mr. Ye?
Yeah, Chair, Senator. We have. Actually, seizures is one of our key core competencies, and we use our brain waves to test drugs if they cause seizures, which is a major issue. And for those that aren't familiar or live and breathe the life sciences industry, the #1 cause of safety failures in clinical trials is actually neurological safety issues. And so there was a paper in Nature a few months ago that showed that mouse brains just are not the same as human brains. It might not be a surprise to many of you, but it is genetically. So. Mm-hmm. And so this ability to make human-like species-specific models can really answer the questions around seizures and the types of diseases that we're going after. Right now, if I list them out, autism spectrum disorder, Rett syndrome, which is a form of seizure, schizophrenia, various forms of dementia. We just signed a couple contracts in the area of dementia, Huntington's disease, Alzheimer's is something that we see coming forward, and we actually have the ability to make Parkinson's brains as well. So this is truly a platform. With regards to vaccines, we worked with US AMRID, that is the US Army Infectious Institute for Diseases. They look at weaponizable viruses, and they've used our brain cells actually to infect our brain cells with Ebola, dengue, Zika virus, EEE, We just reached out to them a few days ago to see if we could be helpful in looking at the toxicity profiles of potential drugs to treat coronavirus. And you can use our models the other way around, not just as an infection approach, but also find vaccines by making using our model as an inhibitory model. So giving the vaccine first to our cells and then introducing the viruses and seeing if it can inhibit infection. So yes.
So, Madam Chair, thank you very much. That was an interesting follow-up. Seemed logical when I see the human brain. But this is more, I would gather, preclinical. You still need human actual trials, but it kind of expedites that initial thing to narrow the sorting out process to drugs that are valid. So you don't have this super, super long time and then find out it's not really working?
Absolutely. Chair,
Ye.
Senator, you're absolutely right. This is really about compressing preclinical time. It can take years in animal testing before you actually get to human trials. If you look at phase 2 statistics, 55% of the time in phase 2, drugs fail due to not being effective. And 30% of the time in phase 2, it is unsafe. So you're dealing with a massive failure rate in phase 2, and by that time, the company has already saved or spent hundreds of millions of dollars to even get to that point. So the ability to, if you will, create a clinical trial on a plate of phase 1 or phase 2 would basically be a paradigm shift and a transformation to the whole industry.
Thank you, Madam Chair.
Senator Kiffmeyer.
I would say, considering that you did a lot of this thinking while you were very sick, goes to show you had some good brain activity yourself. And we are grateful for that. And sometimes when you're flat on your back and you have to, just thinking of some of the good that has come out of that. So commend you for being so productive even while you were so very sick. Thank you.
Further questions? I want to thank you. Could you, on the slide you showed the plate, could you show us how big that plate actually is?
Sure, Chair. It's about a little bit larger than your large iPhones or phones that you have in your pockets. So it's an international standard plate, and we fit about 400 brains or hearts or pancreases in each one.
And Mr. Ye, you had shown me, I believe, a slide where you grew a tumor.
Yes.
A brain tumor, so that that could be used for testing effectiveness of chemotherapy or gene therapy.
Yes, Madam Chair. We had, a lot of us are touched by cancer, not just me, but parents have passed away from cancer. So one of the questions we had is, could you introduce tumor cells into these wells or little petri dishes on a plate to basically mimic metastatic active cancer attacking healthy cells? And we were able to do that in a concept. We took glioblastoma, brain cancer, and introduced those tumor cells into the same well as a single mini brain. And we figured out a way to keep both alive long enough for the tumor cells to infiltrate and attack the healthy brains. And the corollary to this in the animal world is you have to humanize a mouse, you have to inject tumor cells on the back and see it grow. And I think there are ethical issues with that, but instead of killing thousands of mice in a single study, you can take some skin a few months later, have a plate of brain tumor cells that you can screen thousands of compounds. And I'll say that the question that a lot of people ask next is, well, is there a personal precision medicine approach with such technology? And really, there's nothing in the way of us doing that technologically.
Thank you, Mr. Ye, for your time. Thank you, Mr. Patrick. And now you have an overview for us?
Yeah, I just wanted to, just a couple of thoughts as we close here. Madam Chair and Senators, today's presentations were just a brief glimpse into the innovative, changing work that goes on in Medical Alley every day. Their work isn't easy, quick, or inexpensive, but as you heard, it's life-changing. Ensuring work like this continues to happen here will require thoughtfulness from all Minnesotans, but especially policymakers like you, to ensure that Minnesota continues to provide an environment for all health technology and care organizations to succeed. Thank you, Madam Chair and Senators.